Yes, so the chapter 11 is about biotechnology, one of a new concept for you all. And the chapter 11 regarding biotechnology is mainly focused on discussing the principles and processes. Now, principle here mainly refers to the different rules, different laws, or different doctrines that we are going to discuss related to biotechnology. Okay, in simple statement we can say that principle is scientific theorem or scientific law or scientific concept or scientific idea behind any major operation. Okay, so principle here can be simply uh, understood as a concept, an idea. Okay, so principles are another principle here is another term for rule. It can be also called as law, or it can be also called as doctrine, and these are scientific law, doctrine, okay, or simply the scientific background behind any concept is principle. So we are going to discuss in this chapter about the main idea, main concept, main principle, rule or laws of biotechnology. Moreover, we'll be discussing about processes. So processes here refers to a step-by-step event, right? So processes are step-by-step events. Any activity is having step-by-step events, right? So suppose say like if you are going to your school, then there are so many processes, right? First you have to go out from your hostel or home, and then you have to walk. After walking, then you'll be reaching the school gate, and after reaching the school gate, then you'll be walking and then entering into your class. So all these are processes, step by step events, which are mainly aimed to achieve an activity. So that your aim is to visit your school and to reach your classroom. For that you have so many step by step events or activities to be performed so that major activity can be achieved. So in this chapter we are going to discuss mainly about the principles or idea of biotechnology, and we will be looking for different step by step processes which are involved in uh, biotechnology. I hope every one of you are now in the class and following all my instructions. Shall
Yes,
we?
sir.
Yes. So I'll proceed further without asking you all. I'm just checking in just to know that whether my internet connection is proper or not. So we'll proceed further and discuss about the chapter. Okay. So this is the main concept. Now, in this chapter, before understanding the principles, main idea of biotechnology and the processes of biotechnology, we have to understand what is biotechnology, right? Before knowing biotechnology, many of you are now going to, after graduation of class 12, you'll be opting for different professional courses, you'll be going for different degree courses, you may be competing for competitive examination. First we have to understand what is biotechnology, right? And before knowing biotechnology, we have to understand what is biology, right? Bio refers to life and logi refers to study. So study of life, and life here refers to a characteristic feature of living organism, right? And we all living organisms are having different processes happening in our body and many things are happening. So in just what I would say is biology is study of different living organism. So which can be also known as biosciences when you go for higher studies. Biology can be also called as biosciences, or some schools, some colleges, some university may use the term life sciences. And our biology can be categorized under natural sciences. So these are similar terms which are used for biology. Now, after knowing the biology as a subject, as a branch of science, we can now look at what biotechnology is. In term biotechnology, you are having bio here means life, or indirectly to the living organism, right? So we are using the concept of biology, or will be using microorganism or other living organisms in order to come up with some techniques, in order to come up with some technology. And in a way we are now having a new branch of biology or new branch and latest branch of biology called biotechnology. Now we can explain biotechnology in different ways. Okay, first just have an understanding about the background knowledge of biology, biotechnology. So biotechnology became branch of biology in the 20th century, right? We are now in 21st century. In the 20th century, in the end of the 20th century, there was development of biotechnology. And now in all the universities, in all the colleges, we are having a branch of biology which can be studied as biotechnology, right? And it is having good scope, right? So why there is emerging of biotechnology or emerging of this new profession or subject, that is mainly because of it is changing our life. Biotechnology has been changing our life through production of different biotechnological products, right? So in a way we are having improvement in the health. Say like you may have heard about different medicines, different say like you may have simply heard about the insulin which is used for treating the diabetic patient, which is also a product given by biotechnology. Moreover, most of the medicines, vaccines, all these are produced with the help of biotechnology. So there is improvement in health. And again, there is also application of biotechnology, and we are now having a good amount of food production, quality food production, and even quantitative food production. So in a way there is changing if we speak in terms of the lifestyle of ourself. Okay, so biotechnology has become a new branch of science mainly because of there is lots of contribution given by biotechnology. Simply in terms of ourself, we have improvement in health and food production, right? Now what are the different subjects or topics which are being studied by biotechnology, regarding which, as I have already spoken a bit, there are different techniques involved, and those techniques are using live organisms. So microorganism may be used. Earlier we have discussed about the chapter on microbes in human welfare, and that chapter is mainly discussing about different products which are produced with the help of microorganism. Those are also application of biotechnology. And we also have so many other techniques where we are using microorganisms to produce many other food produce. And regarding earlier chapter, we have discussed about processes which are involving use of microorganisms for production of curd, bread, and wine, right? And moreover, we have many other products which can be produced. So biotechnology deals with different techniques, different methods, different strategies for using microorganism to produce different products useful for human, okay, useful for our animals, useful for environment also. Even there is production of enzymes from organisms to produce different products. Okay, so we have microorganisms which are used for production of enzyme, and there is use of those enzyme in order to produce different products. Okay, so enzymology, study about enzyme, is another uh, concept which is studied by biotechnologists. Okay, so in biotechnology we have uh, different techniques which are studied, plus we are having enzymology. Okay, or regarding the techniques of biotechnology, we can even call them as molecular biology techniques. Okay, biotechnology deals with molecular level also. So we also have you also have heard about genetically engineered organism or genetically modified organism, right? We have genetically modified plants, genetically modified animals, we have genetically modified bacteria, genetically modified fungi. So all these are having lots of application. So biotechnology also deals with genetically modified organism. We are also having more of the techniques, say like you have studied about in vitro fertilization technique, which is leading to the formation of test tube baby, which can be also considered as part of biotechnology, as there is involvement of biotechnological techniques. Okay, and even there is synthesis of gene and using it. Synthesis of gene here means synthesizing a new gene. Say earlier coronavirus gene is very harmful, as it is harming the uh, it is making the virus more harmful. But by genetically engineering and synthesizing a new gene which is harmless, now that is making as a vaccine, right? We have DNA vaccine for coronavirus, which is developed in India, is now an application of biotechnological technique, right? And even there is correction of the defective gene. Some people used to born with genetic diseases, and for them we can even have corrective measures with the help of biotechnology. So genetic defective gene or uh, genetic diseases can be even treated with the help of biotechnology. So we are having lots of applications of biotechnology. These are few of the listed techniques, okay? And all these are improving our lifestyle, improving our daily life by improving our health, improving food production, and even we can use biotechnology for improvement and conservation of our environment. Okay, so shall we proceed further? Yes.
Yes, sir.
Okay. Yes, sir. So regarding the biotechnology, I was discussing about lots of application and how biotechnology was first started. Right now, it's important for us to find the definition which is given by European Federation of Biotechnology. Right. So European Federation of Biotechnology had given a definition regarding biotechnology, and this is the statement that they had given. They said that biotechnology is integration of natural science. Natural science means biology, right? We have already seen it, right? So integration means joining. There is application of idea of biology, and there is use of organisms, microorganisms, right? And even use of cells and even parts thereof. Different cells are having different cellular content or different small substances or parts, right? So in biotechnology there is use of concept of biology, and these concepts are applied with using organism, cells, and other substances, so that there is production of different products, and with the help of those products there are different services which are provided. Say like services in terms of production of agricultural produce or food crops, that is improvement in the food, right? And even there is production of different medicines, vaccines for improving our health. And medicines, foods, vaccines, all these are produced with the help of organism, microorganism, or there is use of cells, or there is use of parts of the cell, say like DNA. Okay, there is use of biomolecules like proteins, DNA, and many others. So we can say that according to the European Federation of Biotechnology, they had given this definition, and according to this definition, it says that there is application of biology or natural science along with use of organism or cells, even use of different parts like DNA, other biomolecules, in order to produce different products. Analog means similar. So analogs for products and services. So biotechnology is giving us so many contribution which are usually called as services. Okay, so with this definition we can find both the traditional view of definition for biotechnology, as well as there is also modern uh, idea which is also find, right? So this definition is encompasses this definition encompasses, or it is having both the traditional view and modern view of modern definition for biotechnology. Okay, so this is just regarding the introduction of biotechnology. Now in this chapter, as I had already spoken, there are discussion about the main principles and processes of biotechnology. Okay, so still we have to discuss two of the core techniques, two of the key or two of the main techniques or strategies or methods which are used in biotechnology have to be discussed. So first core or key technique which is used in biotechnology is genetic engineering. So whenever the term biotechnology comes in our mind, then we suddenly came with an idea about genetic engineering and genetically modified organisms, right? And for genetic engineering, we also use the term rDNA technology. R stands for recombinant. Recombinant means re means again and again. Combinant means combination of two or three substances, right? So recombinant DNA technology here refers to manipulating or modifying the DNA or engineering the DNA, deleting, editing, and adding of the DNA is recombinant DNA technology, or it is known as genetic engineering. So regarding the recombinant DNA technology, we'll be having more of discussion in the next chapter, and even in this chapter. Okay, the next chapter, chapter 12, will be about application of biotechnology, wherein we'll be discussing about rDNA technology. And even this chapter, as it is mainly about the genetic engineering, which is the main principle and processes which is involved in biotechnology, we'll be discussing about rDNA technology also. Okay, so I said that genetic engineering or rDNA technology is the method or technique, or it is the strategy where we are changing or altering the chemistry of genetic material. So we are deleting some portion of DNA and adding new parts to that DNA, or we are say like editing the DNA. Similarly, we can even edit the RNA, so that that edited DNA or RNA can be introduced into new host organism, and that host organism now become new or recombinant organism. Okay, and in a way that is having different phenotype means observable characteristic features. You have studied about phenotype in chapter 5 regarding the uh, principles of inheritance and variation, right? So phenotype is the observable characteristic feature, what characters that we observe. So regarding the organism with new edited DNA, we are going to observe that animal as having new features, right? So anyhow, though I'm explaining too much about so many concepts here regarding the genetic engineering, the main point that you have to keep here is we are having two key techniques which are involved in biotechnology, and those two core or main techniques are genetic engineering, and we have maintenance of sterile ambience. Ambience here means surrounding or campus or premises. Whenever we are working on genetic engineering or any technique of genetic engineering, we should try to keep or maintain our surrounding sterile. And sterile here means uh, aseptic condition. Aseptic condition here means our surrounding shouldn't be having any contaminant, shouldn't be having any dirt, or shouldn't be having any of the pathogen or microorganism. So maintenance of sterile ambience here means keeping our surrounding totally clean, which is not having any bacteria, any fungi, any viruses. Okay, that is an important uh, technique, another important technique, or another key technique of biotechnology. Okay, so maintenance of sterile ambience can be also stated as microbial contamination free. Micro contamination means dirtying, right? Once we can say a place is our surrounding or ambience is contaminated if we are having presence of microorganism. Okay, so we should maintain our surrounding micro free from microorganism. Okay, so we can explain the main point which I had explained is also stated here, right? So maintenance of sterile ambience in the chemical engineering process, that means chemical. Why chemical? There is involvement of DNA is deoxyribonucleic acid, which is a biomolecule, which can be also considered as a chemical. So chemical engineering process of DNA should be having totally free environment or totally free ambience, which is not having any microorganism, so that there is uh, proper growth of, suppose if we need to produce one microorganism that can be grown without contamination by other pathogens, by other microorganism, and we can even produce that, produce the product of microorganism in large quantities. So mostly we have discussed in the earlier chapter about production of antibiotics, right? Even there are production of enzymes. Regarding vaccine, we haven't discussed. So we can say that we have two core idea, or two main principles, or two main concept which are involved in biotechnology. These are, first is genetic engineering, which is also known as recombinant DNA technology, and the second one is maintenance of sterile ambience, which is also known as microbial contamination free. Now, hope you all have understood. Can we look at the main idea or the main background of the chapter? Yes.
Yes, sir.
Okay. So let's. Yes. The main idea or the entire chapter can be summarized with this single slide, with the title Principles and Processes of rDNA Technology. So, this is the main idea of the chapter. So, hope you all will follow what I'm trying to explain here. This is the main idea, or if I explain this slide, then you'll be having the main idea of the chapter, and in a way, you'll be having the main concept, and you can read and go through the chapter once, and then you are having all the concept regarding the biotechnology. Okay, so hope every one of you are here. Atishi may be having, say, like some problem, that's why she has left, right? Anyhow, so we said that we have so many principles and processes which are involved in rDNA technology or genetic engineering. So whenever we are going for any of the genetic engineering technology processes, we said that our surrounding should be clean. Our surrounding shouldn't be having any dirt. Moreover, our surrounding shouldn't have any pathogen, bacteria, fungi. Everything should be clean, and then we can start with our DNA technology processes. So first, there is, where's my cursor? Yes. First, we have to isolate the foreign DNA. The This structure, the strand that you are able to see here in the form of helix structure, is representing the foreign DNA. Okay. So first step is isolation of foreign DNA, and we should isolate the vector DNA. The term vector here refers to the carrier. This DNA is going to carry a portion of foreign DNA, and that's why vector DNA is used. Okay, so the vector DNA is usually the plasmid. You are knowing that plasmid DNA are small circular DNA present in bacteria, right? So the first step of biotechnology is isolation. First step of rDNA technology is isolation of your vector DNA and your foreign DNA. Foreign DNA can be from our body. Foreign DNA in the sense the DNA which is foreign to a bacteria. We'll see how it is called as foreign DNA. Suppose this DNA is from human, and if we are requiring gene of interest, or suppose say like if we are collecting the gene for insulin, first we'll be using a restriction enzyme. So restriction enzyme is also like a molecular Caesar. It's not a Caesar, but similar to Caesar, it will function and cut the DNA. And there is cutting of the desirable DNA from the foreign DNA. So this entire length earlier joined, and it is known as foreign DNA, right? Now with the help of restriction enzyme, we have cut, and now we have obtained the desirable DNA. Desirable DNA is also known as gene of interest. Okay, the gene that is required. So this segment, suppose, is the gene for insulin. Now this gene insulin is isolated, right? Same is the case, we have to cut the vector DNA with the same restriction enzyme. Same restriction enzyme will cut the vector DNA, and in a way we are now having, see, two cut DNA. This is small circular plasmid DNA which is cut, and the other one is desirable DNA which is cut with the same restriction enzyme, right? So restriction and regarding restriction enzyme we'll be discussing later. These can be also called as molecular seizure, as we are now discussing about the molecule, that is DNA, a biomolecule, right? And it is cut with an enzyme, and that enzyme works similar to seizure. That's why it is also known as molecular seizure. Here I'm just representing with the seizure mainly to make you understand. Okay, so restriction enzyme helps in cutting your both foreign DNA and your foreign DNA and your vector DNA. So we are now having cut circular DNA, which is called as plasmid DNA or vector DNA, plus we have the desirable gene which is cut. Now after that, we are going to use another enzyme called ligase. Ligase is going to join, it is acting like your glue. It will join foreign DNA or desirable DNA to the plasmid. We have already cut the desirable DNA here on the left side, right? And now we are having vector DNA, which will be joined by the desirable DNA with the help of enzyme ligase. So ligase is like your glue. So suppose say like there is joining of the desirable gene or foreign gene will be joined with the vector DNA. And in a way, we have now synthesis of our DNA. Our DNA here is having our DNA. Why? Newly constructed DNA, newly combined DNA, a recombinant DNA. A recombinant means there is a recombination or reconstruction or there is rearranging of the gene of the vector DNA. Earlier, this desirable gene, that is insulin gene, is not here, right? It is only the vector DNA, plasmid DNA. Now due to addition or joining of the insulin gene or desirable gene or foreign gene with the help of ligase enzyme, this DNA is recombinant DNA, as it is recombined. There is no new combination of the DNA, right? So hope every one of you are following. Yes.
Yes, sir.
Okay, so this recombinant DNA now will be passed into host organism. Host organism which is used in rDNA technology is mainly the E. coli. So we have already seen this rDNA. Now this rDNA will be inserted into the bacteria called E. coli. And this process of uptake or introduction of new piece of DNA into this E. coli bacteria is known as transformation. Okay, transformation is a process in which there is introduction of a piece of DNA. This is what you need to understand right now. In the next upcoming classes we'll be discussing more about the concept. Okay, so we said that earlier this is the bacteria, E. coli bacteria, which is having its outer capsule and envelope and plasma membrane, cytoplasm, and there is a region called nucleoid in which we can find the chromosomal DNA, right? And it is also having its own plasmid DNA. And mesosome, you can see here, infolding of the plasma membrane, right? Now, this E. coli, if we introduce this vector DNA, which is carrying the gene of interest, or this is the rDNA, recombinant DNA, right? Now, this rDNA, if it is introduced into the E. coli, this is the host organism, right? E. coli is host bacteria. So that's why there is introduction of the rDNA, and this process of uptake or introduction of piece of DNA is known as transformation process. So we can understand here saying that though gene of interest, that is insulin, is from our body, from a normal person's body, which is taken out or isolated from the blood, okay, blood sample. But this is now becoming foreign to the plasmid as well as foreign to the E. coli. E. coli is the host organism to which there is introduction of the recombinant DNA, which is carrying insulin. That's why it is called as foreign DNA. Okay, though it's our own DNA, due to new to this E. coli bacteria, that's why it is called as foreign DNA. Okay, now we are knowing that bacteria can multiply, right? Bacteria can undergo binary fission. Bacteria can divide its cell to produce two new bacteria, right? Now this E. coli bacteria will take 20 minutes to divide into two. And while dividing, there is also copying of the same rDNA. See, the rDNA is here in this small cell, new cell, plus there is also formation of the same copy of the rDNA in this E. coli. So these two E. coli can again divide within 20 minutes to form four cells, and these four cells will again divide to form many other cells, right? And such process of multiplication or replication or cell division of the E. coli to form many cells. And in all these cells, we can find the copy of rDNA. They are copy of the rDNA which is being copied, which is being multiplied, or which is being amplified or amplify. The process itself is called as amplification or cloning, right? There is, as the bacteria divides and form two new cells within 20 minutes, there is formation of the bacteria, new bacteria, along with there is copying of the same rDNA into many times. And this process of making copy is known as amplification. Amplify here is indirectly referring to increasing something. So cloning is copying, right? Cloning amplification is process of copying the DNA which is already introduced, and the DNA which is introduced in the mother host cell is having the foreign DNA, that is insulin gene. Okay, now this process of cell division of the bacteria keeps on occurring for say like uh, almost 2 3 days in large tanks, and these tanks are usually called as bioreactors or fermenters. So in these large tanks there is continuous replication or continuous cell division of the E. coli occurs, and there is formation of rDNA copies. Okay, and moreover these rDNA copies will also produce the insulin protein. Insulin gene will be producing the protein, right? And later on we can even uh, kill these bacteria, or we can extract the pure insulin by the process called as downstream processing. So downstream processing means down means the last stage. Downstream here refers to the end of the stream, end of the river. So here in this case, in case of the rDNA technology, the last process is downstream process, and during the downstream processing we are also checking the quality of the product, and the product that we have produced here is insulin gene, insulin protein. The gene from which insulin was produced is insulin gene. So here bacteria can also produce insulin protein from the insulin gene. Okay, so this is the entire process which is involved in biotechnology, and this is the diagrammatic representation given in your textbook. I hope you all had followed. See, in your textbook diagram, not so clear information is given. There is isolation of foreign DNA and vector DNA. Okay, vector DNA here is plasmid. Now both the foreign DNA and vector DNA are cut with same restriction enzyme. So same restriction enzyme cutting both the foreign DNA, that is gene, suppose that is the DNA which is present in our body and it is containing the insulin gene. So the foreign DNA is cut, same case, same restriction enzyme is used and cut the vector DNA. C-cut portion of the foreign DNA and the vector DNA. And these two genes, the desirable gene is joined here with the vector DNA with the help of ligase enzyme. So two enzyme that we have discussed is restriction enzyme. Okay, that you should remember. Restriction enzyme cut the foreign DNA and vector DNA and forms cut portion. And these two cut DNA can be joined with ligase enzyme. And in a way, there is formation of rDNA. See, there is formation of newly recombined DNA. This DNA is carrying desirable gene or the foreign gene, that is insulin gene, suppose, along with the vector DNA, and hence this is called as recombinant DNA, newly constructed. newly combined, right? And that our DNA should be transformed into bacteria. That DNA should be introduced into the E. coli bacteria, that is the host organism. And hence that process is called as transformation. Once the cell of the E. coli divides, then there is division of the rDNA, copying of the rDNA, and such process continuously occurs. Then there is continuous occurrence of copying of the cell, plus there is also copying of the rDNA. Moreover, in the bacteria itself, these insulin gene may produce the insulin protein, and which can be processed and which can be obtained at the last stage called downstream processing. And after that we can even test the quality. And this picture is giving you clear information about how bacterium, first there is isolation of plasmid DNA from the bacterium, right? And it is cut with the same restriction enzyme, and there is joining of human insulin gene. Human insulin gene is obtained from human cell that is present in the blood, or from other cells it may be taken out. So if human insulin gene is combined with the vector DNA, which is cut with the restriction enzyme, and now this is your rDNA. rDNA is introduced into the host bacteria, that is E. coli, which is genetically modified bacterium. And this bacteria undergo process of cell division in large tanks called bioreactor or fermenters. And after that there is also production of insulin proteins by the insulin gene in by these bacteria. And at last there are downstream processing where there is purification of the insulin or extraction of the insulin, and insulin is now ready for distribution and marketing and use in the hospital. I hope you all have followed what I had explained so far. Yes.
Yes, sir.
So the gist of the chapter is all about the principles and processes of genetic engineering or rDNA technology. So tomorrow I'll again discuss the same animation to make your concept clear. Okay?
Okay, sir.
Yes, sir.
I think if I take more of your time, I think you all are now tired. Before leaving that, I shall I explain once about the concept, main concept. Yes?
Yes, sir.
Yes. See, we were saying that in the chapter on biotechnology principle and processes, the chapter number 11, we are mainly having the idea of rDNA technology or genetic engineering concept, and which is having principles, the main background idea or concept of genetic engineering, plus many processes which are involved. And here in this chapter, we are going to discuss mainly about this slide. Okay, say like there is isolation. You have already done experiment about DNA isolation. So you are isolating DNA from plant sample. So here we have isolated this DNA from human blood sample or human body, okay, from the human tissue, and that DNA is called as foreign DNA. Okay, so first step is isolation of foreign DNA, and simultaneously we are also isolating vector DNA from bacteria. And that vector DNA, the vector DNA is usually the plasmid DNA. Plasmid DNA are small circular DNA present in the bacteria, right? So after isolation of vector DNA from bacteria and isolation of foreign DNA from human, okay, now these two DNA are cut with the enzyme called restriction enzyme, and restriction enzyme will cut DNA. Okay, these are acting similar to Caesar. That's why they are also known as molecular Caesar. So after cutting the foreign DNA, then we are going to get the gene of interest or desirable gene, that is insulin gene. And same is the case, we are cutting the vector DNA with restriction enzyme, and we are going to obtain cut plasmid DNA. Okay, now after that we are going to use an enzyme which will join the desirable DNA to the cut vector DNA, and that enzyme is known as ligase enzyme. So ligase enzyme will join the foreign DNA or desirable DNA with the plasmid DNA. So see, desirable DNA joining with the uh, plasmid vector DNA and forms recombinant DNA. Now this is recombinant DNA having the gene of interest, that is insulin gene plus the vector DNA. Now, this rDNA, newly constructed DNA, will be transferred into the E. coli bacteria, which is the host organism. And such process is called as transformation. So transformation is introduction of piece of DNA. Piece of DNA here is the rDNA into the E. coli. Now E. coli bacteria will multiply to form two bacterial cell which are also having the same copy of the rDNA, and the bacteria is also copying themselves, and again after 20 minutes there is formation of few more bacteria. These bacteria will again act as parent bacteria and form many other bacteria. So such process of formation of millions and thousands of bacteria, which is occurring in large tanks in the industries, biotechnological industries, called as fermenter or bioreactor, will be achieved, and that process of copying of number of DNA, number of rDNA in the bacterial cell through the is achieved with the process of amplification or cloning. Cloning and amplification means copying of the piece of DNA, that is rDNA, that is rDNA containing the gene of interest. So such process continuously occurs for 3 to 4 days, and then finally we can extract the insulin gene. While the bacteria started growing in these large tanks, they will release insulin from the insulin gene. Okay, and in a way we can extract the insulin from the last stage of the rDNA technology, that is downstream processing. Downstream means end of the stream. Stream here means river, right? End of the stream we are having downstream. Similarly, here at the end of rDNA technology, we are having downstream processing, wherein we are trying to extract and purify the product, that is insulin, and finally quality testing is done and uh, uh, marketed, and it is sent to the hospitals. Okay, so this is the main concept. If you just focus on this simple diagrammatic representation, it's fine. And even by referring my notes, if you just go through these pictures, you will get good understanding. And in this chapter, the main discussion is all about here in this slide. Okay, this is the diagrammatic representation which is discussed again and again. How what is restriction enzyme, what detail about restriction enzyme, and information about ligase enzyme, process of transformation, construction of rDNA. All these will be discussed. So the main theme here is this entire picture. Okay, so thank you all. Have a good day. So sorry for taking more time. I've taken even your break time, right? So sorry for that. Thank you. Thank you all. Thank you. Thank you all.